Hey, my name is Josiah Zayner, and I have a PhD in molecular biophysics and biochemistry from the University of Chicago. After that, I spent a couple years working at NASA doing synthetic biology. I'm trying to help astronauts be able to recycle waste for sustainability in space. But what I found was that working in the government and academia, I was never able to do the science and research I wanted to do. I was always restrained and held back. I was forced to do science that would get grants or publish papers, and these aren't the things I wanted to do. So, after working at NASA for a few years, I decided to leave, and I became a biohacker. What's a biohacker? Well, that's complicated, but generally the definition of biohacker is somebody who is doing science outside traditional environments. A biohacker is somebody who's trying to explore the world without a degree, without a commercial or, straightforward lab, sometimes working in a garage or shed or their kitchen. But a biohacker can be anyone, and I think that's what differentiates a biohacker from a scientist, is a scientist generally is required to have degrees. A biohacker doesn't. After I started biohacking, I started a company, which we're at now, the company The Odin, and the goal of The Odin was to teach other people how to do science and genetic engineering in their homes. And I want to show you a bit of that today. I want to show you the things that people are capable of, the things that biohacking inspires people to do, and why biohacking is the future of science, I think. Here's the thing, is that science limits the access to information and the people that can do things, and biohacking totally changes that. One of the things that we are trying to make the real world, the biological world, successful and relevant in people's lives. So what I want to show you right here, this is where we grow bacterial cellulose. What we did was we isolated a strain of bacteria from the environment that what it does is it grows cellulose. Cellulose is a material that's found in cotton and plants. This material then can be dried and turned into textiles. So over here you can see examples. Some of this is a little harder. Some of it's a little softer. It's almost like a leather, right? You can see and tell that this is like a leather-like material. Now you could grow this just from a single bacteria in sugar and water. We want people to be able to explore the scientific world, not just for, the idea of getting grants, but also for sustainability reasons and to be able to make cool things that have never been made before. Now you can grow these biomaterials in containers as large as you want, and you could see the biofilms forming in each of these. Now we provide people with kits that allow them to do this, but anybody can do this. Anybody can isolate these bacteria and organisms. The other thing we're really interested in is fungi, mushrooms. Mushrooms have so many cool properties. Not only are they just beautiful, and you can create sculptures and art with them, they're edible, and some of them even have cool properties like glow in the dark. We offer courses and classes here at the Odin that teach people how to grow fungus and use it as a material, as a food, or just as something cool. Because who doesn't like glow in the dark fungi? Next, what I want to show you is where we do our work with animals. Well, it's not really animals like most people think. We do keep fish and a really cool little animal that helps science a lot, and it's called a nematode. C. elegans. Kind of horror. I can't even pronounce the name. You know, some of these Latin names are pretty crazy. It's like kind of horribus elegans. Everybody just calls it C. elegans, but it's a little nematode, and I want to try to show you these little nematodes if I can on these plates. So we take the nematodes and you put them on a plate, and they live off of bacteria. You can see on here, you can see the bacteria growing. That is the yellowish stuff. Now, why do we work with nematodes? We work with nematodes because they are an animal, but they're so small and they're so easy to reproduce and work with. It's crazy. We actually use these little tools, worm pickers, that we made ourselves just out of 30 gauge wire that was clamped to make it like a little spatula. The cool thing about the science that we do is that we try to make it super accessible. We try to figure out how cheap and handmade. So if you look at this, this is just a little cheap bamboo stick skewer that you'd use for food, and a little piece of wire that's stuck in the bamboo skewer in order to pick up worms. Now companies sell these little worm picks for upwards of $50. Right now we're working on helping these nematodes grow without all the traditional materials that are required in fancy labs. What we have over here is we have this inexpensive $15 USB microscope that was purchased off of Amazon, and this microscope, you can actually watch the worms. It's pretty crazy. And people usually use really expensive microscopes that aren't necessarily required. So let's see if we can get some of these worms on the microscope. By using microscopes, inexpensive microscopes and things like that, it makes these materials accessible to so many more people. All right. Take plate. And it's surprising how well just inexpensive stuff works. You know, a lot of times we think, especially in science, people are like, ooh, you need really expensive materials and equipment, but it's totally not true. You don't need expensive stuff. A lot of times you can do this stuff with really inexpensive materials. So here's a little nematode worm crawling around, and you can see this little inexpensive USB microscope. I don't even know if I'd call it a microscope. It's probably more of a camera that you can plug into a computer. Any computer can look at these worms. It costs $15, and most people use expensive microscopes. Now, this is what we are trying to do. This is what biohacking is learning how to do science on the same level as academic scientists, just with a lot less money and resources, trying to get more done with what you have. The next thing I want to show you is I want to show you human cells that we're growing. Now, a big part of biomedical research is being able to grow and using human cells. If you want to be able to test any sort of drug or chemical or genetic engineering, the thing that you want to be able to do is you want to be able to work with human cells first. And so what we've done is we've tried to make human cell growing and engineering accessible to anyone. It sounds like a crazy thing, I know. And if you would have told me that this was possible, or somebody was trying to do this 5 years ago, I would have said it's impossible, right? Usually you need really expensive and fancy equipment to grow human cells. People use CO2 incubators. So what people normally use is they use a CO2 tank that's connected to an incubator, and the amount of CO2 in the incubator is regulated so that the media has the correct pH. It's buffered by the CO2. Is that there are certain medias that you can make and use with human cells that doesn't require CO2. So this CO2 regulator is not actually connected to the incubator. It's completely turned off so that these cells are grown in the absence of CO2. Now, I want to show you some of these cells. So these are some human cells I've been working with. Now, these human cells are actually what are called primary tissue culture. That means they came directly from a person, right? And they've had no genetic modification. So a lot of the cells that people tend to work with, like HEK cells or HeLa cells, those are the cells that people are probably most familiar with, they're actually really messed up. They've been genetically modified to live longer, and they have a lot of abnormalities around them. So when people are trying to look at how cells actually work in culture, like normal cells, what they use is they use primary tissue culture. So let's look at some primary tissue culture cells. These cells are from like human skin, muscle tissue. They actually look really cool. So let's look at these cells over here. So we've been able to grow these cells, really advanced biomedical techniques, without using any expensive or fancy equipment. Again, this incubator that we're using, you can use something really inexpensive, as small and inexpensive as a chicken egg incubator. This microscope is the most inexpensive microscope we can find. It's generally considered a toy microscope, but we're using it in this case to do advanced biomedical research, which is super cool. So over here on the screen, you can see the cells. So the cells you're seeing here are actually human cells that are growing in culture. Let me get this big flask. We got lots of cells. Now what we do with these cells is we grow them up and genetically modify them and test out different biomedical research things. We also provide them to other people and teach other people how to use and grow them, right? There's a limited number of scientists in the world right now, and if we have more people doing and working with human cells, then the world is going to be a better place. People are going to be understanding medicine, and we're going to have so many more drugs that weren't normally accessible to people, right? We need more scientists and genetic engineers in the world being able to study these cells. Look at these human cells that we have right here. Some of them are really interesting and weird, and it's really cool to be able to work directly with human cells. So these are primary tissue culture cells, and we also teach people how to work with like standard cell lines. So standard cell lines like HEK293 cells are used in a lot of biomedical research because they're so easy to work with. They've been immortalized, and these are HEK293 cells right here. So you can see in here, we can grow them in plates and we can genetically modify them. This is an experiment I've been doing, genetically modifying the DNA of these cells, trying to test whether a mutant gene expressed changes the morphology or metabolism or behavior of these cells. An actual human disease mutation. And this is the power, is that like inexpensively, somebody can do something like this, right? This equipment costs just a few $100s, and we sell it to people. We offer classes that are both online, remote, and in person that teach people how to do genetic engineering in their own home, in their own kitchen. And that's the crazy thing, is that the animals we're working with, like C. elegans, or even fish that we've worked with, or frogs or other animals, this technology is accessible to people, right? The world is changing, and biology is the future. It is the technology of the future. And people are going to be experiencing and using biology more than ever. And so it's very important that people understand and learn how to use biology, because in the next 10 years, people are going to see and experience biology like they never had before. You have to understand that, like, this time in human history right now, we have the ability to not only read the genetic code, but also program the genetic code. Program the genetic code of organisms, almost any organism, right? So we are capable of programming living, self-replicating beings. That's crazy. We are capable of genetically modifying humans. That's crazy. And we here at the Odin are trying to give people access to this technology, right? Because if this technology is monopolized and used and owned by very few people, then people are going to be able to control what this technology can be used for. They're going to be able to control who gets to be genetically modified. They're going to be able to control what organisms and who gets to use them. And if we're talking about DNA, the most powerful technology we know of, people controlling the use of that world is going to be very difficult. And so, as a biohacker, we believe that this technology belongs to everybody, and we're trying to bring it to everybody. And even if you're not a biohacker, even if you're somebody who's just interested in technology, you have to understand that biology is the future, and it's going to happen. This stuff is already happening. People are genetically engineering organisms, human cells, animals, in their kitchen, in their garage. Nobody can stop it anymore. And so you can either be part of this revolution or just let it pass you by. But genetic engineering is here to stay, and biohackers are changing the world as we see it right now.